All right, welcome to the next section of Chapter 8, which is all about carbohydrates. In this lecture, we're going to be discussing glycoproteins. So just as a reminder, in previous lectures from this chapter, we talked about the general functions of different types of carbohydrates, and we went over the different structures and functions of some monosaccharides, disaccharides, and polysaccharides. And now we moved on to the last section, which is all about glycoproteins. And we're going to use a few different terms here that might be a little bit confusing to keep them separate, but they are all each distinct individual things, and you should be able to tell me the difference between all of these three different things, which are glycosylated proteins, proteoglycans, and peptidoglycans. So let's get into it. Let's think about, first of all, what is a glycoprotein? So a glycoprotein, defined very simply, is just when you have carbohydrates and proteins that are combined together in one single molecule. And so glycosylated proteins, proteoglycans, and peptidoglycans are all examples of this that we see. So in this picture on the left, we have a glycosylated protein. And so you can see here we have a lot of alpha helices and some loops showing us protein part of the structure. And up here we have some kind of polysaccharide that is another part of this single structure. So we call this a glycosylated protein. In the middle, we have proteoglycans, and we're going to discuss a couple of specific examples of proteoglycans that we see in the cartilage in our bodies. And last, we'll discuss, and that's shown in the middle, and then on the right, we'll discuss last what are peptidoglycans, which is the really important component of the bacterial cell wall. All right, so let's get into it. So why do proteins often get glycosylated? Why are saccharides attached to proteins? Well, we observe in nature that most proteins that are either secreted by a cell or that live on the cell membrane surface of a cell, those proteins tend to be glycosylated. And one big reason for that is, if you think about it, any protein that's secreted or that lives on the membrane is going to be always exposed to an aqueous environment. And so if that protein is glycosylated, as we talked about, saccharides are extremely hydrophilic, right? They have a lot of hydroxyl groups, they're very soluble, they love water, and so these types of secreted membrane proteins are going to also love being in water if they have a lot of sugars on them, right? So where do we tend to see saccharides attached to proteins? They're often attached on the surface, the two loops or terms. And we can see an example of that in this picture on the right. So the protein is shown in blue and the saccharide is shown in yellow, and you can see how it's very flexible. So this is showing it in a number of different potential conformations, and because this saccharide component is so flexible, it's able to sort of cover and protect or hide like a large part of the surface of that protein. And so because of this sort of property of oligosaccharides and polysaccharides, the fact that they're really water soluble and the fact that they want to be on the surface of a protein, this can ultimately help them influence both protein folding and the stability of the resulting protein. So as we'll get into in later slides, some oligosaccharides and polysaccharides are covalently attached to proteins while they're in the process of being translated and folded. And so once you have that really polar soluble sugar attached to a protein, that's then going to influence how the rest of the protein will fold around it. And because of that, it will affect the protein's stability. So if, for example, if you had somehow ended up folding, a saccharide into the center of a protein that's generally hydrophobic, then you'd have a problem in terms of clashing of different molecules with different natures to them, and that would not be a protein's most stable conformation, essentially. So ultimately, what do glycosylated proteins do? What do those oligosaccharides do? They can impact a protein's function. They can interact how a protein interacts with other proteins. They can also impact how cells interact with other cells, because, like we mentioned, a lot of these glycoproteins are on the surface of cells. And it's also what we sometimes see for one protein. We can have one single protein, but we can have variability in what glycosylations you might see on that protein. And those differences where we have the same protein but different oligosaccharides, we call those different glycoforms. So let's get into how we attach those polysaccharides and how we name them and all of that good stuff. So we'll start with O-glycosylation. So we talked a little bit in our previous lecture about glycosidic bonds and how O-glycosidic bonds have an oxygen participating in them. And so O-glycosylations, when we were talking about attaching sugars to proteins, we're going to see those when we have a bond to serine or threonine, because if you recall, both serine and threonine have a hydroxyl group on them. So this will help them want to make an O-glycosidic bond. And the location on the protein of where these types of O-glycosylations are really depends on the protein structure, because like we said, really these can happen on the protein's surface. So if you have a serine or a threonine that's sort of buried somewhere in the protein, maybe not all the way in the center, because you might not see such a polar residue in the center of a protein, but if somehow you have a part of the protein that's not really on the outside or on the surface, then you probably wouldn't see an O-glycosylation there. If you do have a part of the protein that's on the surface or facing the outside of a cell, for example, if it's on the membrane, that's where you'd more likely see an O-glycosidic linkage to sugars. And that's what I'm saying when I'm saying that location depends on the protein structure rather than the sequence. So just because you see a serine or a threonine, you have to think more carefully of where is this falling in the tertiary or quaternary structure of the protein. So what are the most common sugars that are added during O-glycosylation? We usually see a repeating a disaccharide that is made of a galactose and an N-acetylglucosamine. And they have a beta-1,3 linkage, and that's pictured here at the bottom. So on the left, we have this galactose, and it's beta, right, because our N-ammeric carbon has the hydroxyl group pointing up, and that N-ammeric carbon is carbon number 1 in our sugar. And it's linked to an N-acetylglucosamine. So we have our original glucose molecule that's been modified by this on the 2 carbon by an amine that's been further modified by an acetyl group. So that makes our N-acetylglucosamine. This N-acetylglucosamine is alpha because the N-ammeric carbon has the hydroxyl group pointing down. But the linkage that we refer to is to carbon number 3 on this molecule. So the linkage is not at the N-ammeric carbon for this N-acetylglucosamine. So overall, we would call then this bond a beta-1,3 glycosidic linkage. And it's an O-glycosidic linkage. And so this basic disaccharide that we see here can be further edited by glycosidases and glycosyltransferases. These are enzymes that can modify sugars by cutting them or adding more sugars on. And the addition of these O-glycosylations is always post-translational. So this is after a protein has been completely synthesized and folded and has been sent over to the Golgi apparatus. So an O-glycosylation might have less of an effect on protein folding because the protein is already folded by the time we add on this O-glycosidic linkage. And hence that's why its location really depends on the protein's structure, because the protein is already folded when it receives an O-glycosylation. So let's talk now about N-glycosylation, similar but different. So N-glycosidic bonds we know don't have an oxygen in the middle, right? They have a nitrogen. And so we'll see N-glycosidic bonds happening for glycoproteins when the protein has an asparagine residue. But it's not just the fact that there is an asparagine there. We need what we call a sort of consensus sequence. So it's 3 amino acids in a row. The first one is asparagine. That's where the actual glycosidic bond goes. The second one is any amino acid, as long as it's not proline. And then the third amino acid could be either serine or threonine. So we need those 3 amino acids in a row in the protein sequence in order to form an N-glycosidic bond. And as part of N-glycosylation, we actually have the addition of a 14-sugar oligosaccharide. That's the beginning of the N-glycosylation process, and once we attach on that 14-sugar oligosaccharide, it gets further edited, again, by those enzymes, those glycosidases and those glycosyltransferases that will add and subtract sugars to that oligosaccharide. And so N-glycosylation also differs from O-glycosylation because of where it starts. So the initial addition for N-glycosylation is co-translational. It happens in the rough endoplasmic reticulum. So while a protein is being translated, it's being sent into the rough endoplasmic reticulum. And inside of that compartment is where we have that first addition of that oligosaccharide. And so the protein is still being translated, it's still in the process of being folded, but that's where the end glycosylation starts. And then once the protein's translation and most of its folding is complete, that protein will also go to the Golgi apparatus, where the full editing of the end glycosylation will be completed. But like I said, it starts out in the rough ER, and even the editing of that oligosaccharide also begins in the ER, but it's not done until the protein has finished being processed in the Golgi apparatus. And so because of all of this different locations of where this glycoprotein can be edited, and because it's happening sort of during translation and folding, we can get this really huge heterogeneity and a whole big variety of different types of end glycosylations. And so let's look into that a little bit more detail now. So here is sort of a diagram of the process of end glycosylation. So we start with that 14 residue, oligosaccharide, and that gets attached to this asparaginine residue on a protein. And that 14 residue oligosaccharide is composed of a number of different sugars. The key is up here. So at the bottom here in brown, we have N-acetylglucosamine. Then these blue inverted triangles we have here are mannose. And then these orange diamonds we have are glucose. And part of the editing process begins by actually cutting down a lot of the sort of top part of this oligosaccharide. And all of the oligosaccharides actually ultimately will get trimmed at some point to this 5-sugar unit, for lack of a better word. And this core, right? And this core is actually, we see it in all end-linked oligosaccharides. But once it's broken down into that core, it then gets built back up by adding a variety of other sugars. And that's where all of this variability comes in. So we have this core pentasaccharide that we see here that I'm circling. And then on top of it, those enzymes can add all different kinds of sugars like galactose and sialic acid and N-acetylglucosamine and fucose and all kinds of variety of stuff that we see here. So that gets us into a little bit more about immune responses and immune recognition of ourselves and things that are not part of ourselves. So a lot of antigens, which are molecules that are recognized by antibodies, are glycoproteins. So they have a protein component and a sugar component. And so as we've discussed in previous chapter, antigens, we know, are cell surface molecules that will stimulate an immune response. And generally, the only antigens that stimulate an immune response are non-self antigens. So during the process of development, B cells that make antibodies that recognize antigens on our own bodies are deleted. Only B cells that don't recognize us are allowed to continue living. But the antigens that our immune system recognizes, as long as they're not from us, they can stimulate an immune response. And so they can be either on other types of cells or other types of proteins and sugars that we don't have in our bodies, as well as microorganisms, which we think of as more sort of the traditional stimulus for an immune response. So one interesting example of this, where you have antigens on other cells that are mammalian, just not in yourself, are blood types and blood group antigens. And so the A, B, H, and Rh antigens that are present on some but not all red blood cells are really important for helping us determine whether or not someone's blood is compatible with someone else's blood when they need a blood transfusion. So let's start talking about the different blood antigens. So you probably heard of blood types A and B, and you're probably looking at this table going, what on earth is H? So H is basically the basic blood group antigen. So all red blood cells make this H antigen. And so no one is ever going to make an antibody against the H antigen because all of our red blood cells make this H antigen. And it's composed of a galactose that's attached to an N-acetylglucosamine as well as a fucose. And the A and B antigens, and I don't remember if I said this, but this is on the surface of red blood cells. And so when we have our A and B antigens, they're actually adding on to the H antigen. So the A antigen, it has that same base that we see in the H antigen, as well as the B antigen, right? These are exactly the same, galactose, N-acetylglucosamine, and fucose. But the A antigen, in addition, has this N-acetylgalactosamine, and the B antigen, in addition, has this galactose. And so this is a really small difference, right? It's only one single sugar, one single monosaccharide that's making this big difference in between these different types of blood group antigens. And this can have massive clinical consequences. If you transfuse someone with the wrong blood type, you will get hemolytic anemia, and it may not end well. So let's get into why that happens. So you've probably heard of, maybe, you might even know your own blood type, but there are 3 main blood types that we'll usually use for classifying blood transfusions, and this is A, B, and O. So first let's look at what it means when you have this blood type and what antigens you have on your red blood cells. So if we look at the first row of this table, if you're blood type A, your red blood cells will have the A antigen, as well as the H antigen. If you have blood group B, you will make the B antigen. If you have blood group O, you don't make the A or B antigen, you just have that base antigen, the H antigen. How do those different antigens arise? It's because different people who have different blood types have different glycosyltransferase enzymes that will add different sugars on to that H antigen. So if you have blood group A, your glycosyltransferase adds an N-acetylgalactosamine. If you have blood group B, your glycosyltransferase is adding a galactose. If you have blood group O, you have an inactive glycosyltransferase. And then, of course, if you have blood group AB, which I skipped over, you make both the A and B antigens. And this is because you have 2 different copies of glycosyltransferase, and each one adds one of each of those sugars. So you will end up with both. And as we talked about, when B cells that make antibodies that recognize antigens are getting developed, you'll make all kinds of B cells that recognize almost anything, and then you'll delete the ones that you see in your own body. So let's start at the top of this chart again. So if you're blood group A, you will make antibodies against the B antigen. You won't make antibodies against the A antigen because you're expressing it, right? You won't make antibodies to the H antigen because that's part of the A antigen. If you have blood group B, you're going to make antibodies against the A antigen. And if you have blood group AB, you're not going to make antibodies against either one because you have both of them in your own red blood cells. And then finally, if you have blood type O, you will make antibodies against both the A and B antigens because your red blood cells express neither of those antigens. So what does that mean in terms of what it looks like inside of our bodies? Here's the sort of another chart that is showing how that all works in a more pictorial form. So we have here again our 4 main blood groups. We have group A, group B, group AB, and group O. So group A and group E are each expressing the A and B antigen. Group AB, those red blood cells will express both antigens, and group O just has that plain old H antigen on it. And so on the next row, we have then the picture of the antibodies that each of those people in each of those blood groups will make. And so something interesting you might notice about these antibodies is that they're all arranged in groups of 5. These types of antibodies are called IgM. And when our immune system makes antibodies against saccharides, against sugars, they tend to be of this type, the IgM type. When we're making antibodies against proteins, like we discussed earlier, that's when we'll see IgG, as well as some of the other types of antibodies. And so, in case you were wondering, that's why they're in groups of 5. That's because they're IgM, and IgM is always like this. They're attached together in groups of 5. And so anyway, for each of our blood groups, they make the antibodies against the group that they are not. So in group A, you have anti-B, group B, you have anti-A, group AB, you have none, group O, anti-both. And so this is really important for determining which blood types you can receive in a transfusion, right? If you're making anti-B antibodies, you cannot receive type B blood because the antibodies in your own body will then attack those red blood cells and cause them to lyse. And so if you're in group O, you're also in trouble, right? If you receive either A or B or AB, you have antibodies against all of them. So you cannot receive any other type of blood if you're group O, except for other group O blood. If you're in group AB, you're lucky, right? Because you don't make any antibodies, so you can receive any type of blood because you don't have antibodies against any of them. So, all right, well, that moves us on to another type of antigen, which is the Rh antigen. So there are actually several different types of Rh antigens, but the one that we most frequently are thinking about when we're talking about the Rh antigen is the Rhd antigen. This is the one that's most likely to induce an immune response. But even in the rhesus D antigen, there are still a lot of different types of mutations in it, and you can also have it deleted, and it doesn't affect your health in any way. So there are some people have the rhesus antigen present, and some people have it absent, and so that's really what we're looking for when we're looking at is it there or is it not there? And that will give you, so if you have the rhesus antigen, that's what gives you a positive type blood type. So you probably heard of a blood type as being, O negative or A positive or AB negative. And this means so if you're positive, you have the rhesus antigen. And so because you have it in your own body, you don't make antibodies against it. If your own blood lacks the rhesus antigen, the Rhd antigen, then you have a negative blood type. And then because you don't have it in your own body, you're going to make antibodies against the Rh antigen, which again can cause a problem for blood transfusions, right? If you yourself lack the Rh antigen and you receive blood that contains it, the antibodies that you have in your own bloodstream are going to then attack those red blood cells and cause them to lyse. And again, that is going to be not helpful for you. So, and then one other difference between the Rh antigen and those other carbohydrate antigens we were talking about before, the A, B, and H antigens, those were all carbohydrates. They're sugar-based. Our antibodies against them are IgM. The Rh antigen that we see here is a protein antigen, and so the types of antibodies we'll make against it will be mostly IgG. So now that we've thought a lot about these different blood types and the different immune responses we might make against them, take a moment and think what blood type would be considered the universal donor blood type and why. And please go ahead and pause the video for a few moments and think about that, and then we'll talk about it in just a moment. So the answer is O negative, and we already sort of discussed why. So O negative blood, that means your own blood lacks A and B antigens, as well as the Rh antigen, right? It's got no antigens. And so anyone receiving it, it doesn't matter what antibodies the recipient has, because O negative blood doesn't have any of the antigens that might stimulate an immune response. So hopefully that all makes sense to you, and I believe we're going to move on from there, talking a little bit more about protein glycosylation in general. So again, I want to stop and think for a moment about which of the following statements are false regarding protein glycosylation. So again, please pause the video for a moment and think about that, and then we'll talk about it in a few seconds. Okay, so let's start with the first one. So N-linked oligosaccharides are attached to asparagine and lysine residues. So what's the part here that's false is lysine. N-linked oligosaccharides are only attached to asparagine residues. So let's consider question P. O-linked oligosaccharides are attached to serine and threonine residues. That's true. So let's consider question C. N-glycosylation begins with the addition of a 14-sugar oligosaccharide. That is also true. So let's now move on to question D. O-glycosylation begins in the rough endoplasmic reticulum. That's false, right? So which one begins in the rough endoplasmic reticulum? That's N-glycosylation. So N-glycosylation starts while the protein is still being translated into the ER. Editing also begins in the ER, and then editing is completed in the Golgi. O-glycosylation, like is stated in this question, begins and ends in the Golgi. It does not happen in the rough ER. So finally, let's consider question E. Addition of N-linked oligosaccharides may determine how a protein folds. And yes, that's true. And we just sort of explained why. It's because end-linked glycosylation begins in the rough ER while the protein is still being translated and in the process of folding. So if you add an oligosaccharide, it's going to want to be on the surface of the protein, and so it might affect how the protein might have otherwise folded without that glycosylation. So let's move on to talk about some other aspects of protein glycosylation. So one really interesting example of this is cartilage. So cartilage is a type of connective tissue that we see. It cushions our bones, that it's frequently found in the joints. And it's a type of tissue that is firm but also flexible. And part of what makes it like this are its components, right? So it's made of three main components. The first one is chondrocytes. Chondrocytes are cells that are going to produce collagen. So the cells are little factories that make collagen and then secrete it outside of the cell into the cartilage. Then the next component of cartilage is collagen. It's being produced by chondrocytes. Collagen itself is a type of protein, and it's very fibrous, and it makes this meshwork throughout the cartilage. And in this picture on the right, we can see collagen fibers shown in blue, forming this nice meshwork. And then the third component that we have in cartilage is proteoglycans. Proteoglycans are a highly hydrated gel matrix. And in this diagram, they're shown in this kind of pinkish red color that look almost like feathers or brushes. That's interwoven all throughout the collagen. And something to note here, in the cartilage, there are no blood vessels. So for a lot of our tissue, we need blood to carry nutrients into the tissue and waste away from the tissue. But in the case of cartilage, there are no blood vessels. And that is because the mechanical properties of cartilage itself actually help to promote the diffusion both of nutrients in and of waste out. And part of this is because of those viscous and elastic properties of GAGs that we've talked about in the previous lecture, and we'll go over that a little bit in the next slide. So let's talk about those proteoglycans. And they have, like I mentioned, this kind of brush-like structure. And why is that? It's because they have a number of different components. So first, that sort of main backbone of a proteoglycan is a GAG. And here in this example, we have hyaluronate as an example of a GAG. And it's very, very long, right? It's made of many, many repeating subunits. And attached to there, we have this core protein. In cartilage, the name of that core protein is called aggrecan. And the core protein is attached to the hyaluronate non-covalently, actually. There is no covalent linkage. It's all through other types of interactions. And the linkage is through this, what we call a link protein, that's not actually pictured here in this diagram. The next really important component are these small end-linked oligosaccharides that we see over here on the right that are attached to the hyaluronate. And then attached to those end-linked oligosaccharides, we have more GAGs, right? We have keratin sulfate and chondroitin sulfate. And these are O-linked GAGs, or glycosaminoglycans, in case you forgot. And recall we talked about how keratin sulfate and chondroitin sulfate are extremely charged, right? They have all these negatively charged sulfate groups as well as negatively charged carboxyl groups. And so overall, they love water, right? And so molecules like this, these proteoglycans that we're looking at here, really, really love to take up water. They love to interact with water. They love water. And their weight when they're wet is massive compared to their weight when they're fully dry. Because of this reason, they just love to take up water and interact with water. And this is the other reason we have that that property we were discussing before, where they're easily compressible, and as you compress them, you can squeeze out water. So in the case of cartilage, if you compress it and squeeze out water, you can also remove any waste that's dissolved in that water, being carried along by that water. But eventually, if you squeeze them too hard, then all of those negative charges will start to repel each other that we see, for example, like all those sulfate groups we were just mentioning. And so once you have all those charge repulsions happening, then we'll see that elasticity property where the proteoglycan will want to return to its original shape. And this way maybe it can take in different water or fresh water that might have different nutrients or different components dissolved in it or carrying in it. And so that's this property that helps to promote diffusion of waste and nutrients both in and out the cartilage, as well as giving the cartilage the ability to cushion your bones and your joints. And so, like we mentioned, proteoglycans are these huge complexes that contain hundreds of core proteins, and they respond to pressure by squeezing out water and then pulling it back in, depending on the pressure and the charge-charge interactions. So that brings us to our next example of a glycoprotein, which is peptidoglycan. So just remember, very important to keep these separate because they're very different things. Proteoglycans are what we see here, this example in our cartilage. Peptidoglycan is what we're seeing here in bacteria. And peptidoglycan is the major component of the bacterial cell wall. So why do bacteria have cell walls, right? So one really simple answer is cell walls help to give shape and stability to the bacterial cells. And also they're another barrier that protects the bacteria from the environment. So if there are antibiotics or other components in the environment that might be harmful to the bacteria, the cell wall is just one more layer that helps to keep that kind of thing out from the bacterial cell. Cell walls also have components in them that are really important for virulence. So they can help a bacteria invade its host, or they can help sometimes to try to hide from the immune system when they're invading. At the same time, your immune system can also recognize components of the bacterial cell wall to help recognize when you see a bacterial infection in your own body. And that goes along with this fourth point, which is that the cell wall contains antigens. So there are antigens that are on the surface of the bacteria contained in the cell wall that antibodies in your immune system can then recognize and use to help eliminate those bacteria from your body. And pictured here on the right, we have two main classes of bacteria. We have gram-positive bacteria and gram-negative bacteria. So the thing that they have in common, right, is they have a cytoplasm that's surrounded by a plasma membrane. And then on the outside of there, it's a little bit different. So for gram-positive bacteria, shown on the top, right outside of that plasma membrane, you have a really thick cell wall made of peptidoglycan. For gram-negative bacteria, outside of the plasma membrane, which is shown in yellow, there's a space called the periplasmic space. And then just outside of that is the cell wall, shown in purple, but it's much thinner. And then in addition, outside of that cell wall, they have a second outer membrane. So that's really the main component, the main difference between gram-positive and gram-negative bacteria. Gram-negative bacteria have an extra membrane outside of their cell wall, and they have a thinner cell wall. Gram-positive bacteria have that super thick cell wall because that's really their main protective barrier, aside from the same plasma membrane on the inside. So what are the components of bacterial cell walls? It is peptidoglycan. So peptidoglycan is a polysaccharide. So the main repeating unit in there is a disaccharide made of N-acetylglucosamine, pictured here on the left, which you've seen before, and it's attached to an N-acetylmuramic acid, which we haven't, I don't think, seen before in these slides, but again, it has that same, it's yet another monosaccharide, right? And we have this linkage here, so we have our anomeric carbon with the hydroxyl group pointing up, so it's beta, so we have a beta 1, because it's on the first carbon. On our muramic acid, we have the linkage is to carbon number 4. So we have a beta 1,4 glycosidic linkage in between the N-acetylglucosamine and the N-acetylmuramic acid. And those are beta 1,4 linkages. Attached to the N-acetylmuramic acid, we have this little peptide over here that has some amino acids we've seen before and some relatively unusual ones. So when we were discussing amino acids a couple chapters ago, we were talking about how most of the ones we see in nature are L amino acids. And so here we see two examples of those, L-alanine and L-lysine. But in addition to that, on the bottom of this diagram, we also have a D-alanine. And then we also have this interesting example of an isoglutamyl. So this is also an amino acid. It's a glutamic acid residue here, but the attachment is different. So let's think about tracing our amino acid backbone. So our L-alanine, we have the amino group, the alpha carbon, and the carbonyl. For our lysine, we have amino group, alpha carbon, and carbonyl. For our D-alanine, we still have amino, alpha group, and carbonyl, alpha carbon and carbonyl. Our isoglutamal looks different, right? Our peptide backbone is actually, or what is normally the peptide backbone is like this, right? We have our amino, our alpha carbon, and our carbonyl right here. And the attachment for this peptide bond that attaches this isoglutamal to the L-lysine is actually the part of the side chain from the glutamate, the carboxyl group that we see there. So that's why this is not just a regular glutamate residue, it's an isoglutamal. And that is that little peptide chain that I just described, and it is covalently bound to the polysaccharide over here. And the other third main component of the cell wall that we'll talk about is called a pentaglycine bridge. And we can sort of help to visualize that by looking at this picture over here. So shown in this diagram in yellow and orange, kind of making these long lines. So that's that long polysaccharide that's made of N-acetylglucosamine and N-acetylmuramic acid. And then these little greenish circles that we see throughout, those are that peptide chain that we just described that has the standard and non-standard amino acids. And then that third major component is the pentaglycine bridge that we see shown in purple going across, kind of connecting the strands of the polysaccharides and peptide chains to one another. And those pentaglycine bridges are just what it sounds like, right? It's 5 glycine residues. And those are going to help connect those peptide chains to each other. That helps to make this whole very relatively rigid structure that ultimately will help to form a very protective cell wall. And something really interesting about the peptidoglycan is that it's not just linear because of all of the different components, and it will naturally form a coil that we see sort of zoomed in over here. And this coil actually wraps around the bacterial cell. And so it's not just like a flat sheet like you might be imagining. It actually is a coil, which helps make it really thick with extra sort of layers in it. So let's think more about the cell wall. Cell walls are a really great target for antibiotics. So when you think about antibiotics, these drugs that help to fight off foreign invaders and microbes, the best antibiotics are going to be drugs that attack only the microorganism that's invading you and not any kind of cells in your own body. And we just talked about how bacteria, right, have cell walls, but mammals like us, we don't. So it's really great if you can have an antibiotic that's going to specifically target the cell wall, because that drug will only hurt the bacteria that are inside of you and not your own cells. So two types of antibiotics that we'll use as examples here. The first one is penicillin. So penicillin is a drug that's going to inactivate the cross-linking enzyme that helps to connect together the peptidoglycan. And then there's also vancomycin. Vancomycin binds to the peptidoglycan precursor and prevents it from being built up. So these are two different antibiotic drugs that both attack the cell wall in some way. And so essentially, if you have this drug in the environment that is preventing the cell wall from being built up, it basically is going to stop bacteria from being able to divide, because every time a bacteria divides, the daughter cells need to build new cell walls, and if you have antibiotics present in the environment, they can't do it. So, but bacteria are also trying to live their lives, right? And so in some circumstances, bacteria are able to both develop and to share antibiotic resistance. So recall that bacteria divide very rapidly. They live their lives very fast and they divide a lot. And so this can sometimes result in a lot of small mutations and small changes in their genomes, as well as in some of the proteins that their genome codes for. So if they're in an environment where they're in the presence of an antibiotic, like a low amount of an antibiotic, there will naturally be some bacteria in that group of bacteria that maybe already are a little bit resistant to that antibiotic. And those bacteria will be selected to survive compared to some of the other bacteria that are living with them that are not resistant to the antibiotic. And so the few bacteria that are resistant will then ultimately be able to keep growing and dividing in the presence of an antibiotic. And sometimes those antibiotic resistance genes that a bacteria has or can develop, sometimes they're located on plasmids, and some bacteria are actually able to share plasmids with each other. And so sometimes you'll see bacteria in an environment that has antibiotic in it, and those bacteria will actually be swapping and sharing some of those antibiotic resistance genes with each other. On the other hand, a lot of the antibiotics that we have are actually developed from microorganisms, because bacteria are also competing with themselves for the same environment sometimes, or competing with each other. So you might have two different types of bacteria who both want to live in the same type of environment. So one of those types of bacteria might make an antibiotic that selectively targets the other one, so it can try to fight it off and keep its environment to itself. So those are all sort of maybe hopefully interesting things that you can think about when you're thinking about antibiotics and cell walls. So let's talk about now mechanisms of resistance that some bacteria have against both penicillin and vancomycin. So some bacteria are able to make an enzyme called penicillinase or beta-lactamase, which inactivates penicillin. Essentially, penicillin has a functional group in it called a beta-lactam. That's really important for its function in inactivating a cross-linking enzyme during peptidoglycan synthesis. And penicillinase, this resistance gene that some bacteria have that makes this enzyme called penicillinase, is able to then inactivate penicillin. Similar but different, some bacteria are actually resistant to vancomycin. And so bacteria that have resistance to vancomycin have a gene that basically codes for using a different precursor to help make the cell wall, so that they no longer need this original peptidoglycan precursor that is bound by vancomycin. So now at this point, I just want you to take a moment to recall to yourself what are the major functional and structural components of the three big topics that we talked about in this lecture: glycosylated proteins, peptidoglycans, and proteoglycans. And I will leave you to think about that for yourself, and we'll go over it in class. And with that, you should be ready now to answer that question I left you with, as well as the Chapter 8 glycoprotein practice questions.